told we want to keep everything on time, and thank you all for taking time out on this cold, blustery morning to join us here. Some of you have been to this event before, our myeloma symposium, and some of you have not. And so I just wanted to tell you a little bit about the origins. My name is Natalie Callander, and I'm a professor of medicine here and have had the opportunity now for 19 years to be here at UW putting together our myeloma program. So I want to tell you a little bit about the origins. But first, I think we have somebody who'd like to say hello to you, and that would be our head of our cancer center. This is Dr. Howard Bailey. So thank you, Howard, for stopping by. We'll give you a round of applause.
My apologies for making you wait and Dr. Callander wait, but again, my name's Howard Bailey, and as the director of the Carbone Cancer Center, I just want to thank you for your attendance, for your willingness to get involved. We are very, very proud of the physicians, the providers, the researchers you're going to hear from today. We have literally one of the best myeloma programs in the country, thanks to the people that you're going to hear from today. And you're a big part of that with your interest, with your support, with the ongoing participation that many of you have in the different studies. So on behalf of the Carbone Cancer Center, welcome. And thank you for your interest and involvement, and I hope you have a great morning. So again, thank you.
Thank you very much, Howard. All right. Okay. Well, again, let me go back for people who may not be familiar with our myeloma community. And so back in 1999, before a thing like patient advocacy was really around, 3 individuals here in Madison, their pictures are there on the right, Jane Buffett, Charles Koval, who was a professor of emeritus of entomology here, and Nancy Schultz decided that what they really needed was a patient group for myeloma, because at that time there really wasn't a lot of treatments for myeloma, and they were looking for information and for ways to become educated. But what they did is they went further than that, and a few years after, they decided to found the Trillium Fund. And basically, this was money to be used to promote clinical research and scientific research in multiple myeloma. Now, this is now its 20th anniversary, so it's very fitting we're here today. And I got here at the end of 2004, and in 2005, just a few months after I got here, I was asked to present to a group. At that point, it was about 10 people, about what was going on with multiple myeloma at UW. And really, there was very little. But I want to sort of walk you back in time with some of these statistics. So back in 2005, we had a few treatments for myeloma. These are abbreviations for things like melphalan and prednisone that were pills that we used back then. This is something called vincristine, adriamycin, and dexamethasone. And when you look at these response rates, this was any chance of lowering your myeloma protein at all. We're not talking about complete remission, perhaps only with transplant. But the more sobering part is over here, that back in 2005, survival for people with myeloma was measured in months. And so 2.5 years was considered great if somebody made it that long. And occasionally we saw somebody living longer than that. But the situation was pretty grim, maybe a little bit better if you had a stem cell transplant, which was something really new back then. But what we did in 2005, when we had really nothing going on, is we were able to open 2 trials that year. And one was a trial that was led by a national group here called ECOG, where we were looking at lenalidomide, which was experimental back in 2005, and using 2 different types of dexamethasone dosing. One was the traditional Mayo dose, where they used to give 40 milligrams 4 days per week, every week, if you can imagine how much steroids that is, or 40 milligrams only a week, which still seems like a lot, but that was the norm at the time, that higher dose. So we were able to join that study. And in addition, we also opened a trial that was going to look at using an autologous, using your own cells transplant for myeloma versus a donor transplant. The idea was we were curing leukemia with donor transplants. How come we couldn't do that with myeloma? So what we found out from both of these trials is a couple of things, is that the higher dose of dexamethasone was much more toxic. And even though it looks like people responded a little bit faster, at the end of the day, people who got the lower dose in green here, they lived longer and had many fewer complications. And in addition, as far as the transplant trial, we learned that an autologous transplant, shown here in red in these curves, did better with many, many years of follow-up versus an allogeneic transplant. Now, one of the things that's so amazing about this, and is just a testament to what's happening, we have people who are on both of those studies who are alive now, which is fantastic. So it's in many ways, we have lots of good news to tell you. So if you look at what's happening with myeloma survival every decade, going back to the 1980s, we continue to improve survival across the board. And part of that is newer drugs that we've had. But now, since 2020, the survival is increasing by about 1.4% per year, which is amazing if you think about that. And transplants down here in the right corner have something to do with that. It has really helped people live longer. But the other thing that we had some concern about is, let's say you're over age 75, were you benefiting from myeloma research? And the answer is now yes. So even if you are our oldest patients in your 80s, our oldest person on a clinical trial here happened to be 94, you are benefiting from innovations, and we hope that that continues. So we have had the privilege here of having many studies in the last 18 years that I've been here. Some of these have really changed practice, but the question is now, where are we? So one of the goals, we have three really, have been, we want to have a clinical trial for every stage of myeloma. So whether you're at the very beginning, or maybe you've had myeloma for a number of years. We also want to figure out how to minimize side effects. We're doing better at controlling myeloma, but we don't want people to have their quality of life continually worsened by side effects of our treatment. And finally, we want to be innovative. So in terms of earlier lines of therapy, there are people who have what's called smoldering myeloma, and that's a phase where people have protein, they have cells in their bone marrow, but they actually don't really have any symptoms from it. But we know that a fair number of these people, at least in what we call the highest risk, half of them are going to go on and get myeloma in a couple of years. So I'm very proud to be the national principal investigator on this trial that is investigating modest treatments using lenalidomide or lenalidomide plus daratumumab for 2 years in the hopes that we can not potentially cure it, but keep them from getting active myeloma. And you may say, well, what's the point of that if you're not making the myeloma go away completely? Well, as we're talking about this, the longer you stay in the game, the more likely it is that we can come up with something down the road that may be curative. And so we think that this is a very reasonable way to go for people who don't have any symptoms at all from their myeloma. And this may turn out to be practice changing. We also know that people with myeloma who have PET scans, I'm going to show you, this is one from a person. These PET scans are used now all the time to document myeloma. This happens to be one from a person. All of these spots are active sites of myeloma, and these little yellow dots are myeloma too. But we also know that people with smoldering myeloma can have a little bit of fuzzy pink here, meaning there's some activity, but not myeloma itself. So we're looking at imaging in this particular trial to see if it can help us figure out ahead of time who's more likely to get myeloma or not, and intervene then appropriately. So we're also looking at ways to both intensify treatment, but also limit the time that people are on it. And so there's two examples of this that we have going on. One is, one you may remember, and some of you may be living this, is that back in 2009, a trial that we did here looked at using lenalidomide after a stem cell transplant versus observation. And we're able to show that there's a big difference in terms of controlling myeloma and even some in survival. And it looked like every type of myeloma benefited in these particular trials. So if you had sort of easier to treat myeloma, harder to treat myeloma, you benefited, but not everybody benefits the same amount, certainly if you have harder to treat myeloma. So what we're looking at is, can you make everybody do better? But there's also probably some people on these trials who didn't need to just stay on lenalidomide forever. They could have stopped. So we're involved in two trials specifically. One is called the DRAMATIC trial that's going on right now. And this is a trial where we're asking people after a stem cell transplant to take lenalidomide, one of our best drugs, or lenalidomide plus the antibody drug daratumumab, and take it for 2 years. If at the end of the 2 years, shown over here, you don't have any disease apparent in a bone marrow biopsy with a technique called minimal residual disease, then you get to stop. Half people will stop treatment, the other half will continue. And we're going to see how this works out with our hope that stopping, if you're in a very deep remission, is very much comparable with continuing. Now, one of the things that I'm very proud of is, and you all know this because you live it, is we didn't have any part of this study when it started to see how people were actually feeling, what we call quality of life. So I'm very happy that I'm the quality of life chair on this nationally to try to find out as people go through this treatment, how they're feeling, not only just side effects, but how they're feeling if they're asked to stop treatment. Is there a lot of anxiety? Are people going to feel very much adrift? So we'll see how that study turns out. 1,400 people nationwide, and we've been very fortunate to be one of the sites here, number 11 in the country, to have people on this trial. We've also been part of what's called the master trial. This is a trial we helped design and execute. And the point here is we decided to take maybe the best treatments we have right now for myeloma. These are the 4 drugs up here: daratumumab, carfilzomib. Again, there's Revlimid and dexamethasone. Have people get pretty intensive treatment for a number of months, including a stem cell transplant. But if you show us on 2 separate occasions that your myeloma is in a deep remission, we're going to stop your treatment and just see how you do. And this particular study has really been practice changing. This study has now been presented nationally, internationally, many times. And what we were able to show is that many people were able to get to that very deep remission with this strategy. Not every person, I want to point out, because we're only at 81%, but this is much higher than many other studies looking at this. But what we also found out is if your myeloma cells have mutations and you only have 1 mutation or 0 mutations in them, you're actually doing very well with this strategy to just stop treatment after you finish, which really concludes in about a year. Unfortunately, if you have more than 1 mutation, it did seem to be that people are starting to show some relapse. So we are going to target these groups in our upcoming trials that we're designing that you're going to hear about a little bit later. And one of the more important things about this particular trial, as many of you may or may not know, multiple myeloma is twice as common if you're African American. And we specifically had a lot of African American patients on this trial, showing that not just a few people with myeloma, but every group benefited from this approach. So now we're going to talk a little bit about cellular therapy, which is really going to be a game changer for all hematologic disorders, we think. But specifically how this is done is to take cells, in this case lymphocytes, out of the body, and those cells are then actually, in most cases, infected with a virus that is carrying genetic information to reprogram them. The cells are grown and multiplied, and then they're re-injected. And these cells themselves become a living drug. So you are no longer taking medicine every day. You are actually having these cells in your body fighting the cancer, with the hope that eventually we'll get to the point where these cells could last for years. Now, this bottom circle here is showing you, we're going to talk specifically about one target that's being used called B-cell maturation antigen. But these targets down below, there's many of them in myeloma, and we think we're going to see CAR T transplants develop to hit all of these. Now, we've been also very, this is hot off the press, this draft, but we've been very happy to be involved in what's called the KarMMa-3 trial that offered these CAR T transplants to people who had had somewhere between 2 to 4 types of treatments. In general, they had not responded well to those treatments, and they were asked to either have a CAR T transplant or go through standard chemotherapies. In this case, one of the nice things about this study, and one of the reasons we selected this, is if you were asked to go through the standard treatments, you could do what's called crossover and go get a CAR T if your chemo didn't work. Now, what this study has showed us preliminarily is that if you are asked to get the CAR T, this was a much better strategy. 3 times the control of your myeloma versus standard chemotherapy. And the important thing to remember as all of these months are going by, people are on no chemotherapy at all, which is really, really something that our patients who are on this study very much appreciated. And we think what's going to happen, and I'll show you another example of this, is CAR T transplants may move up to the very beginning and may replace an autologous stem cell transplant in just the next couple of years. We're also involved in what's called the Target CR trial. This is something we also helped design. And this is a trial where we're asking people if they've been through a stem cell transplant and had a few months of lenalidomide, but not looking like they're in a complete remission yet, then we're asking them to go through a CAR T transplant at that point. And this is a pilot trial, meaning only a few people are going to be asked to do this, about 40 nationwide. And we are fortunate to be one of the sites here. Patients get this CAR T transplant, and then we are asking them to take 1 year of lenalidomide afterwards, but then stop treatment. And so we think that this is going to be a model going forward that people are going to copy in their trials, and again, may set a new standard of care. And we're very, very happy to be able to offer this to people. So I want to show you just what has happened in our myeloma community since the time I've been here. So these are just the number of individual people with myeloma that we see every year now, with over 2,000 this year and still counting. So what does that mean? I think that means several things. I think one thing that people worry about when they see something like this is, does that mean myeloma is just way more common? We do think that there is an increase in incidence worldwide in multiple myeloma, so that may be one part of this. But I think there's two other things that are even more exciting. And one is that all of these people down here, they're living to here, so that we get more and more patients because everybody is living longer and they're here to contribute to this, which is wonderful. And the second thing is that based on what you're going to hear today with some of our research and some of the trials I've told you, we're really turning into a place that people with myeloma want to come, preferentially for treatment, which is what we want to continue. And we are still a big transplant center. Now, these numbers have fluctuated a little bit, double what we used to do. They fluctuated a little bit because of COVID. There was some concern about doing transplants then. This number may continue to go down, actually, if we end up doing more CAR T transplants at the beginning. But we are very, very happy to be able to be a big part of the myeloma community, not just here in Wisconsin, but nationally and internationally. So another accomplishment is, and if people were here before, you may remember that maybe I would be up here by myself as the clinical person, but just in the last couple of years, we're very excited to have 3 new dedicated myeloma physicians. This is the future. So Dr. Schmidt coming to us from Emory, Dr. Gahvari, who was trained here, went to San Diego, and some bizarre reason decided to come back to weather like this. And then Dr. Matthew Brunner, and you're going to hear from Dr. Schmidt and Dr. Gahvari. And then, of course, we have our wonderful translational partners. Dr. Miyamoto is going to talk to us. He has addressed you many times, and he's going to talk about his work. And then we're going to introduce to you Jing Zhang, who's going to tell you about a very exciting model that she's working with. So I'm going to turn the podium over to Dr. Schmidt now and let him take over.
All right. Does that sound okay? Okay. Okay, so I am super excited to be here to talk to everybody, both here in the room as well as online, and to kind of move forward into just talking about newly diagnosed multiple myeloma and what are we currently working on and what are we looking to for the future in terms of our clinical trial and research program here at UW. I've got a couple of disclosures. These are more just a few companies actually think I have something useful to say, and as well as some of the companies that are supporting some of the clinical trials here that I'm involved with. So before I get into the science and the research, just for those of you who don't know me, I'm Tim Schmidt. As Dr. Callander mentioned, I came here from Emory, where I did my fellowship, but I trained at a variety of different places before that. And I started here right at the beginning, kind of early phases of COVID, which is, of course, a great time to come to a new place and meet people. But I'm still very excited to be here. I grew up in St. Louis, Missouri. I'm married to my high school sweetheart, Shelly, and she has graciously moved around the country with me as I've gone through this. Outside of work, I'm a big sports fan. I do apologize. I'm a Cardinals fan, but I actually won't apologize for that. But I also have some teams up here in Wisconsin that I'm happy to adopt. So in terms of the treatment landscape for newly diagnosed multiple myeloma, we have generally a few different phases. So we start with what's called induction therapy, where there's a lot of cancer cells in the body, and we're really trying to minimize that as much as we can. And we use combinations, so typically 3 to 4 different drugs to bring the body from a lot of cancer down to very little, sometimes even achieving remission. For some patients, we will go forward with an autologous stem cell transplant, as Dr. Callander was alluding to. But everybody eventually, we can't do this intensive therapy forever. So we eventually go to a maintenance strategy, which is less intensive, but it's still something. It's still some sort of treatment to keep these low level of cancer cells under control for a long period of time. Inevitably, though, at least right now, we know that patients are still going to relapse. And second line and beyond therapy is something that we are still talking about. So how can we improve upon this? And some of these Dr. Callander already mentioned, but first is we could kill more cancer cells up front. And by doing so, maybe we can help to prevent those relapses. And this involves identifying better treatments. And also, even among people who respond, it's important to identify those patients who, even though there's been a response, might be at a higher risk of relapse. And maybe we should be doing something more for these patients. The second area that we should improve upon is minimizing treatment and eliminating those side effects when we can. All of these things that we do have potential side effects. And so I think that one of the things that's necessary to be able to achieve this second goal is the first objective is having better treatments might allow for us to back off on the back end. But also identifying some patients who, after they've gone through their therapy, they might be at low risk of that early relapse. And could we potentially do less? So I'll go through a few different phases of ways that we could achieve some of these goals. And so the first, potential way is, can we give more therapy to get rid of that residual myeloma after initial treatment? And so this goes into a type of therapy or an idea that has come into play called response or MRD adapted therapy. So what is MRD? And this is a fancy medical word that's measurable residual disease. And so the concept for this, I think the best analogy that's been described is that of an iceberg, that when somebody comes in with newly diagnosed multiple myeloma, there's a lot of symptoms. This is what we see from the myeloma. It's what's there on the surface. But the reality is that beyond that, there's a lot more disease that is under the surface that we just can't see. And so even with our initial treatments, when we start to see the monoclonal proteins and everything coming down, and we get that reassurance that there's a response, even when we get to the point where somebody might be in remission, which is this complete response, there's still myeloma left over, which is why we still see relapses even among patients who achieve remission. So why does it matter to try to go deeper and find those lower levels of disease? And it generally speaking, because the more likely we are to find myeloma, the more likely it is that somebody is going to relapse. On the contrary, if you can't find any evidence of that myeloma under the surface, that means patients, generally speaking, are less likely to have a relapse. So just kind of general principles about these curves, which we throw up here on these graphs all the time and may not do as much explaining. But essentially, these are graphs of patients who are involved in clinical trials. And this data comes from a trial that happened in Spain. But as these curves go down, this is somebody who relapsed. And when they looked at this trial, according to conventional criteria, a CR or an SCR, these are patients who achieved remission. And we saw that those curves did not go down as much as those who didn't quite yet achieve remission. But when they took a closer look at this and looked to see, even among these patients who were in remission, could we find disease under the surface? And if we couldn't, these were patients here in this MRD negative graph. And it starts to look like if you can still find that disease under the surface, these are patients who are still more likely to relapse than those where we can't. And so we're starting to incorporate this discussion of MRD into a lot of these newly diagnosed trials and using this as a factor to try to say, should we be doing more or should we not? So you're going to hear a lot about this trial, which hasn't yet started, but we're really excited about it. And Dr. Callander mentioned MASTER, which I think maybe, I think to put it mildly, was an innovative trial. I think in reality, this has been looked at as a really exciting, potentially practice-changing trial, and we're excited to be a part of the second development of that. So I'm going to talk about a couple of components of this later, but essentially this trial is for patients with newly diagnosed myeloma. They're going to get a 4-drug induction regimen, and after 6 cycles of that, we'll check for MRD. And for those who we can still detect that MRD, then we're going to go on to explore 2 different strategies. One would be transplant and then a 2-drug maintenance regimen, and the other would be incorporating this drug called teclistamab in combination with daratumumab. So what is this? Dr. Callander mentioned CAR T, essentially harnessing the immune system and re-engineering it to go fight the cancer. Well, teclistamab, and there's a whole host of these other drugs, this is a class called bispecific antibodies, where essentially instead of re-engineering the T cells themselves, we are using drugs to take the T cells that already exist in the body and have them go find the myeloma cells. So this is a type of immunotherapy where essentially we're binding to both of these, telling the T cells to recognize those myeloma cells and kill it directly. And without going into a lot of detail about some of the historical benchmarks, but typically drugs in multiple myeloma would get approved if they had a response rate in this late relapse setting of about 30% and to work for 3 or 4 months. But as a single drug, even in today's landscape where patients have gone through 5, 6 lines of therapy, right now, what we're seeing with this drug is a response rate of 63%, most of which were actually in remission. And those patients who responded are responding typically for a year, sometimes more. So we are going to take a look to see for patients who are still MRD after that, MRD positive after that initial therapy, does this combination of teclistamab and daratumumab help to overcome that detectable MRD, drive more people into MRD negativity, and hopefully prolong their degree of remission and survival? For patients not going forward with a stem cell transplant, we have another trial through this group called ECOG, where this is taking a look at existing drugs that we have and how can we potentially utilize these in a smart way to preferentially do more for those who need it and maybe not for those who don't. So we're using a 3-drug regimen of daratumumab, lenalidomide, and dexamethasone for 9 cycles. And then patients are randomized either to continue with that or to add bortezomib or Velcade kind of mid-treatment with the idea that perhaps adding in that 4th drug might improve the survival of patients who still have detectable MRD after that initial induction therapy. And this is even without a transplant. Commander. So this is another exciting trial through the same group that did MASTER and is going to do MASTER 2. And this trial is looking at combinations involving a drug called iberdomide after a stem cell transplant. So what is iberdomide? If you kind of think about it, it sounds very similar to a drug that many people know and perhaps don't love, lenalidomide. And so essentially, here's this cartoon that, lenalidomide, its target, it fits into this nice little binding pocket here. And when it does that, it sets a cascade forward in terms of myeloma cell death. Well, compound 6, as it was called then, which now has been called iberdomide, has that same little piece of it, but it's also got this side chain. Now, this still fits very nicely into this binding pocket, but it actually fits tighter. And so by being more specific, that sometimes means that it's more effective. It also means that it's less likely to get into places that it's not supposed to go, and there might be fewer off-target side effects. So if we can find a drug that's better and have fewer side effects, that's what we're looking for. And so this trial called Commander takes patients who've gone through any induction therapy, a stem cell transplant, and who still have detectable disease by this MRD test. And there's 2 potential options here. One is a combination of daratumumab with iberdomide and dexamethasone for 6 cycles, followed by maintenance with this new drug for a total of 18 cycles, or another cohort that incorporates carfilzomib, which is kind of the 2nd generation of bortezomib or Velcade, looking to see if this new combination can overcome the impact of that MRD positivity. Second way that we might be able to improve upon this is getting rid of transplants. And for anybody who has been through transplant or seen people go through transplant, it's tough. And we know that it improves survival in myeloma, but at the same time, if we don't have to do that, we would like to not do that. So the second part of this MASTER-2 trial that I mentioned, the bottom part earlier, is taking a look here. So for patients who got that 4-drug induction regimen and achieved MRD negativity, undetectable disease with our most sensitive techniques after just 6 cycles of therapy, the question is perhaps we might be able to get away without a stem cell transplant in those patients. So patients in this study who are MRD negativity are going to be randomized either to continue getting that 4-drug combination and then reduce that to 2 drugs, or a stem cell transplant followed by maintenance. And so that's going to help us determine within this specific group of patients, perhaps could we get rid of transplant? And then thirdly here is, can we ever stop maintenance therapy? And this is a question that gets brought up in clinic all the time. I've heard it from several of my patients. And so Dr. Callander already alluded to this trial called DRAMATIC. This is done through a group called the SWOG, or Southwest Oncology Group. So this is taking patients who've been through any sort of induction therapy, a stem cell transplant, randomizing them to either lenalidomide, which is currently the standard of care, or to add daratumumab to that, and to do that for 2 years. After 2 years, we'll check for MRD. And for those who still have detectable disease, they continue with the maintenance that they were intended to go on. For those who are negative and now are 2 years out from transplant, we're then going to randomize patients either to continue that maintenance, which would be the standard, or to just stop therapy, observe, and see whether or not this has any detrimental outcome. And the thought is that if you're still MRD negative after 2 years, perhaps you don't need that continuous maintenance therapy. And then in the MASTER-2 trial, the final kind of piece of this is, regardless of where you get randomized to in this, whether that's to not do a transplant, to do a transplant followed by kind of standard maintenance therapy, or at least maintenance with daratumumab here, or to get the teclistamab, that if you see in those bone marrow biopsies, whatever treatment that you've had, if time after time on 2 consecutive assessments at the end of here, there's an undetectable MRD, we will then stop therapy entirely and see what happens. And hopefully we're going to see that with these really deep responses from some novel therapies, that we're not going to see high rates of relapse. So collaboration here is really key. And I think, this is a big testament to what Dr. Callander has done over the years in terms of getting our myeloma program up and running to where it is, that we are participating in a lot of these cooperative groups, some of which are nationwide consortiums, some of which are just a couple of university programs that decided to come up with innovative trials together. And then also sports brings people together in some way. And the Big Ten even has its cancer research consortium that we're participating in all of these. And through our participation in that, we can also use these trials to answer some other questions. One of which is, what is the importance of these genetic abnormalities that we're finding in the myeloma cells, and what does that mean in terms of their activity? And so even, me getting here within the first couple of months, I was fortunate to be involved in one of these side projects through the ECOG group that led to a national presentation at a national meeting and a publication forthcoming about the impact of these chromosome 1 abnormalities. And then also looking at the MASTER, trial, we were able to look at an entirely different question: is what happens to the immune system when we use all of these therapies? And when we start doing more and more intensive treatment, what impact does that have on our ability to make normal antibodies and have a normal functioning immune system? And so we learned a lot from that trial by going back and taking a closer look. We did learn that eventually the immune system does recover from that. But these are it's just a small sample of some of the things that we can do by cooperating in some of these groups. So in the future, we're certainly looking at doing more of these correlative studies of these cooperative group trials, further exploring. One of the interests that I have is some of these genetic things and risk assessment. One really interesting question that we've already decided that we're going to look at from Master 2 is, can we test for MRD not from bone marrow biopsies, but from the peripheral blood? How great would that be if we can get away from bone marrow biopsies and just use blood tests to figure this out? We still need to study it though, and that's one of the things that we're going to do. We'll certainly take a look at using CAR T cells in the frontline setting, bispecific antibodies, even for patients who are not planning to go through with a stem cell transplant. Always looking at improving the supportive care, quality of life efforts, those things that we can do to help patients and their caregivers get through the days and help to improve people's lives, not just lengthen them, and so many more other things that are yet to be determined. So with that, I'll end. I'll say thank you for your attention. And from here, I believe that we're going to invite Dr. Gahvari down to give the next talk, or maybe it's so thank you for your attention. I think that's what matters.
This is the hardest part for me. Can everybody hear me? Great. Okay, so let's see. Okay, here we are. So I'm Zhubin Gahvari, for those of you who don't know me, and it's my extreme privilege to be able to speak to you today for my portion of this presentation entitled Back to the Future, Relapsed Multiple Myeloma. And so I have no relevant conflict of interest disclosures. And so in the spirit of my colleague, Dr. Schmidt, for those of you who don't know me, I'm originally from Virginia, in that state we shall not name, Illinois. But then after medical school in Virginia, I did all of my training here at UW postgraduate. And on the bottom left is actually a picture of my parents and Dr. Callander at my fellowship graduation. And then kind of apropos to the sudden change in the weather in the last day or two, I spent the last couple of years in beautiful sunny San Diego. And despite all of the nice beaches there, I was actually very honored to be able to come back and rejoin the program here just last year. And one of the goals of my portion of this talk is to show some of the work that we have been involved in at this program, and actually some of the work I was able to witness firsthand during my own training and participate in, which helped really bring my interest in focusing on multiple myeloma for my career. And so kind of to frame the conversation a little bit, Dr. Schmidt focused on newly diagnosed myeloma. My focus is on relapsed myeloma, and particularly getting more somewhat both early relapse, but also late relapse of myeloma. And that's one of the unfortunate stories still right now, is most people's myeloma eventually outsmarts the treatment that it's on, and we have to do something else. And the natural story of myeloma has often been that each medicine combination we use works a little not quite as good as the thing before it, until eventually patients unfortunately run out of effective therapies for their myeloma. And one of the big things about all the clinical trials that we've been involved in is of course trying to find newer and newer options. And so when your myeloma has relapsed, the principles that we all want to follow, and of course, a real conversation about relapsed myeloma is going to take a lot longer than the time we have here, but basically principles include using at least one new therapeutic class that you haven't used before, trying to avoid therapeutic classes to which your myeloma is no longer sensitive. Very importantly, taking into account previous treatment-related toxicities, as well as patient health history and preferences. Some of this is certainly easier earlier on. Given the number of agents we have for myeloma, oftentimes at your first or potentially second relapse, you have multiple agents and choices available, but eventually you start having less and less choices. Very importantly is the urgency of effective treatment. How quickly is the myeloma relapsing? Is this a slow burn, or is this a very aggressive relapse? How quickly do you need a response? And finally, is there an available clinical trial? And the one although I just said each thing we use works not quite as well as the thing before it usually, what bends and breaks that curve is if we get a new type of therapy that we have never had before that attacks the myeloma in a way that it has not really had a chance to develop cross resistance to, then we can completely really give a treatment that controls symptoms and gives a lot longer than we expected to have. And so on that vein, and kind of why I've labeled this talk Back to the Future, is I think going a little bit back in time in some of the newer targets we've had in myeloma in the last decade will kind of frame the future, but also intersects with what we have done here at Wisconsin and what I have personally observed and been able to participate in as being a member of this program. And so this schematic here, this cartoon, shows a number of different targets that are under investigation or have been used to target myeloma. And in the center here in particular, inside the cell, you'll see a number of therapies that you may be familiar with that historically have been the original novel agents for myeloma, particularly proteasome inhibitors and immunomodulators. And more recently, a lot of the newer therapies for myeloma have focused on finding targets that are on the cell surface that are more uniquely found only in plasma cells and hence in myeloma and not in other normal tissues of the body to minimize off-target effects and minimize side effects. And what I want to start with is CD38, a cell surface receptor that has two FDA-approved therapies to target it, daratumumab and isatuximab, both what we call monoclonal anti-CD38 antibodies. And although Dr. Callander and Dr. Schmidt have discussed newer immunotherapies, really, for example, focusing both on B-cell maturation antigen or BCMA or GPRC5D, I would argue that the anti-CD38 monoclonal antibodies are kind of the original novel immunotherapy. I mean, 2015 now seems like a long time ago to some of us, and it's kind of impressive that it's been almost 9 years now. But back in, this was the first kind of these agents that are targeting a cell surface marker that wasn't found elsewhere. And this schematic is really focused on daratumumab, but this is just a quick reminder that this medicine works in multiple ways to both directly and indirectly via the immune system kill myeloma cells. And where this intersects with Wisconsin and the treatment of relapsed disease, we'll get to just in a moment. But the story with daratumumab is, imagine you've used multiple proteasome inhibitors, multiple immunomodulators, you've received a transplant, you've received cyclophosphamide, and you're running out of options. Suddenly, we had a therapy that checked a lot of the boxes for how you want to treat relapsed myeloma. Very well tolerated, minimal side effects, and when you add it to a standard of care agent that you are still sensitive to, you saw significant benefits in terms of how long your myeloma was controlled compared to what was previously available. And here I have kind of shown 4 different studies. On the top left is daratumumab and lenalidomide with steroids versus lenalidomide and steroids. On the top right is daratumumab and Velcade versus Velcade. On the bottom left is daratumumab and pomalidomide versus pomalidomide and pomalidomide dexamethasone. And the bottom right is daratumumab with carfilzomib and dexamethasone versus carfilzomib and dexamethasone. In all of these studies, we're looking at patients with relapsed myeloma, some earlier relapses, some later relapses. They had different number of prior therapies, but in all of them, we saw a similar theme, that doing a 3-drug combination based off this novel agent, daratumumab, showed much longer periods of disease control for myeloma. And so what became natural was, okay, our standard of practice of treating relapsed myeloma changed. If you've never had an anti-CD38 monoclonal antibody, when your myeloma stops being controlled, our practice became you should receive a combination with daratumumab. And more or less universally at relapse, everyone received daratumumab-based combinations based on all of these studies. And so, well, that was a good thing. The problem is once you've shifted everyone to this new therapy, what happens when that new therapy stops working? And so our suspicion was that this was not a good thing, but the truth was 5 years ago, nobody knew. And so in order to better decide or for our patients what the choices were after daratumumab-based therapy stopped working, and to better have a plan and an expectation going forward, we were fortunate here at Wisconsin to participate in a multi-institutional registry study with 14 other academic institutions around the United States, where basically we all had the same question: how do we best help our patients after an anti-CD38 monoclonal antibody stops working? And so we participated in what was called the Mammoth study, and I was very fortunate with Dr. Callander to participate in this work during my training, where we pooled all our patient data together to describe what happens to patients after this new class of therapy that we never had before stops working. And unfortunately, what we saw there was our suspicion was kind of confirmed that it looks like you're reaching the end of the line at that point, where the median survival, and this isn't how long your myeloma is controlled, but this is how long you have left to live after you have no longer respond to your anti-CD38 monoclonal antibody, was less than a year, which was obviously not acceptable. And while in a sense this was not what we wanted to see, this was our moving ground for kind of moving the field forward, because we came to a different definition of what is advanced stage myeloma. Because as you know, some people have myeloma for a few years, some people have it for many years. When does the myeloma become aggressive and harder to treat? Is it when you've had 8 different treatments or 4 different treatments? And we moved the field from the definition of how many years you've had it to more of a functional, relevant definition, which is this term triple class plus refractory, which is if your myeloma is no longer responsive to an anti-CD38 monoclonal antibody, an immunomodulator, and a proteasome inhibitor, that is the new modern definition of late stage myeloma, where based on the standard of care available a few years ago, we don't have many good therapies to offer you. And this told us we need to do better. And so how do we do better? And this is, again, we're looking to the future, not just the past. And so we obviously need more than just an anti-CD38 monoclonal antibody. We need new options, we need new combinations. And so although daratumumab was well tolerated, using it in a multi-drug combination, 3 drugs worked better than 2. And what I didn't really talk about was using it earlier. Patients seem to live longer than using it later. Still, when the daratumumab stops working, you need something else to do. And so for the next parts of the talk, I want to focus on some of these newer options that we think can, again, help bend the curve for our patients and looking to ways to make myeloma treatment better in the future and make our patients live longer. And so for part 2, I'm going to focus on 2 agents. One of them is on the left here is isatuximab, the cousin of daratumumab, which is another anti-CD38 monoclonal antibody. Again, well tolerated and effective like daratumumab. But on the right is an agent that I think has maybe gotten a little lost in the press in terms of all the excitement about new immunotherapy. I think some of us have forgotten about selinexor. And selinexor is a selective inhibitor of nuclear export. And this as you see, this is not a cell surface target. This is an intracellular target. But by targeting this, you can sometimes have selective death of myeloma cells. And what people may forget is selinexor was developed before the pandemic. And in fact, here at Wisconsin, we were very much involved in the development and approval of selinexor as a standard of care for relapsed myeloma. And so on the left is data showing, including data that we participated in the Mammoth study. This curve on the blue is patients who receive conventional available treatment options as of 2018, when their anti-CD38 monoclonal antibody stops working and they're at least triple class refractory. That's their expected survival, but patients who receive selinexor-based therapies did better. And so if you had nothing else available, selinexor was better than your standard of care. And I think that's important to remember, that this is an effective agent even in late relapse myeloma. And furthermore, on the right, we've been involved in a number of studies under the umbrella STOMP, which are studies looking at selinexor-based combinations in relapsed myeloma. And this is one of those subarms, selinexor with carfilzomib and dexamethasone. And what you see here is multiple subgroups, including late relapse myeloma, triple class refractory, as well as high-risk cytogenetics. This is an active combination even in late or aggressive myeloma. And so one of the limitations I feel, and we all feel in some of the selinexor data, is anytime you use an agent late on in myeloma, the numbers aren't going to look as well as you use it earlier on. And so one thing I want you to keep in the back of your head right now is, well, what happens if we could use selinexor earlier in someone's myeloma course? And so holding that thought, just now to isatuximab. Isatuximab, like daratumumab, is very effective in relapsed myeloma. And this is a curve from the IKEMA study showing isatuximab, carfilzomib, and dexamethasone in the blue. And then on the red is just carfilzomib and dexamethasone. And what you see, not only is isatuximab, carfilzomib, dex active in this population of relatively early relapse myeloma, including many patients that were lenalidomide refractory, ISA-KD shows the best numerically PFS that we have ever seen in this space. And so when looking at trials, innovative trials that we can design to help move myeloma care forward, if we have an agent such as selinexor that looks effective in aggressive myeloma but hasn't been studied much upfront, and we have an agent such as isatuximab that is very well tolerated, and we've seen the benefit of 3 drug combinations, why not a 4 drug combinations? And so one of a new trial that we have designed here at UW with Dr. Callander, Dr. Schmidt, and myself, and that I'm very honored to be a part of, is this combination selinexor, carfilzomib, isatuximab, and dexamethasone that we have given the acronym SCID or SCID. And the idea again is, what about 4 drug combinations? Dr. Schmidt discussed this when talking about for newly diagnosed myeloma. And one of the issues is if it works well for newly diagnosed myeloma, we should also study this for relapsed myeloma. And there have been no yet approved 4 drug regimens for relapsed myeloma. And so this is a phase 1b, phase 2 study that we are leading, but we are going to open through the Big Ten Cancer Research Consortium. And so we're very excited to, as we say, sports bring us together, partner with like academic institutions in the Big Ten to have this trial available elsewhere. And so this will be a phase 1b, phase 2, which means that we will have an early 6 to 12 patient arm to sort of make sure we have the correct tolerable dose, but then expand to 50 patients in our phase 2 when we're looking at efficacy. And so although the primary objective is to look at safety of this combination, we are also very interested in looking how well it works. And the reason that I have included this picture from Back to the Future is, as you recall, in the movie, the rock and roll guitar scene maybe was a little bit ahead of its time for the audience. And so using the same theme, we have designed this study to be as innovative and as ahead of its time as we possibly can. And so we are hoping that this is as inclusive as a study that we can make it. And so we have designed this to be available for hopefully almost all patients with relapsed myeloma, including prior B-cell BCMA-directed therapy. And really the only restrictions to enrollment will be refractoriness to the study drugs or recent exposure to them. And the other aspect of trying to make this study as relevant as possible is, as Dr. Callander and Dr. Schmidt alluded to, there are patients who have high and very high risk myeloma who do not get the same benefit from the therapies that we have available that other patients do. And it's that population of patients that needs innovative combinations the most. And so we are intentionally enriching this study to have at least 50% of the population involve high-risk features, including gain amplification 1q, deletion 17p, and double hit myeloma, where you have multiple high-risk genetic features, mutations both present. And along with this theme of being innovative, we are also including several correlative studies, as we call them, or additional sub-studies where we're trying to generate as much practice-changing data as possible from this study. And to quickly talk about some of these correlative studies, One of them is the inclusion of the SKY92 gene expression profile. And so we were very fortunate to meet with the group that provides this gene expression profile, and they're very excited about this study. And as some of you may or may not know, the current risk stratification scheme for myeloma, stage 1, 2, 3, comes from something we call the RISS, or the Revised International Staging System. But as we all know, that staging system is not perfect. And there are patients who have functionally high-risk disease that is not captured by that staging system, who do not do as well to respond as well to treatments as we would expect them to. And my colleague, Dr. Schmidt, for example, has been very involved in broadening the definition of high-risk myeloma with his work on gain amplification 1Q. But we even know that that doesn't cover everyone with high-risk disease. And so another way of looking at the risk profile of your myeloma is through what we call a gene expression profile. In SKY92, it takes RNA from a bone marrow aspirate and sequences it. And on the left is information from a study looking at SKY92 versus the RISS in patients with newly diagnosed myeloma. And what I'm trying to show here, but of course it's hard to see, is this red line here is, or this red and this blue, this purple, I guess you would call it. I'd call it purple. But basically, what you're seeing is that there are patients with high-risk myeloma that aren't captured by the RISS, that are captured by the SKY92 profile, that are doing worse than the RISS would predict them to do. And what we also have potentially seen from the study is by combining the two risk stratification systems, we may have an even better, more precise way of finding patients with high-risk myeloma and better and more appropriately following them for how they respond to therapy. And so this SKY92 gene expression profile has not been validated in a relapsed myeloma setting yet. And one of the aims of this study is hopefully to provide data to move the field forward and show the utility of this gene expression profile in a relapsed population. Another correlative study that we're very excited about is looking at stool. And I know that seems like a funny thing to say, but one of the works that we participated in previously here, and some of you may have participated in, is we did a study by one of our former colleagues, Dr. D'Angelo, who's now at Nebraska and will participate in this study, is taking stool samples from patients with multiple myeloma who underwent an autologous stem cell transplant. And on the left, what we see is that the diversity of the microbiome in patient stool declined from before a transplant to after, and recovered afterwards. And there is obviously a lot of ongoing interest in the gut microbiome and how it affects patients' immune systems. And one aspect of immune health is how your myeloma responds to treatment. And so what this study also showed is that lower gut microbiome diversity was correlated, and that lower diversity is the lower bars here, was correlated with a lower depth of response to autologous transplant. And so we wish to further look at this question of stool diversity, or diversity of the gut microbiome in stool samples, by looking at stool samples before and after treatment on this protocol to see if we see similar changes in diversity and whether those changes correlate with how well the treatment works. And the same way that you know on TV, there's wait, there's more, I have one more that I wanted to add to this. And so one other exciting aspect of this study is isatuximab is currently available only as an intravenous formulation. It does not yet have a subcutaneous administration. And subcutaneous administration is under development, and patients on the study will have actually access to the on-body delivery system for subcutaneous isatuximab, which should be easier and better for our patients. And so it's kind of one additional extra thing we're very excited that will be provided. So, you know, I've spoken a lot about CD38 as a target, both with daratumumab and isatuximab. We've spoken about selinexor as not that old, but people have forgotten this can be an effective target in myeloma, particularly even in late myeloma. And so we've talked about multi-drug combinations. And so I would still be remiss, even though I first was saying, let's not talk about the newer immunotherapies, I also wanted to talk a little bit about the newer immunotherapies. And so in addition to BCMA, another target on the cell surface primarily of plasma cells and located very little elsewhere in the body is something called GPRC5D or G protein receptor, G couple protein receptor 5D. And so, as you can see, it's a mouthful of a word, but basically, this is something that we're very excited about because, in fact, in the last year, you may be familiar with talquetamab, which is a bispecific antibody that targets this and has shown very good effectiveness in late-line triple-class refractory myeloma. But what we do not have is a CAR T yet directed towards GPRC5D. And the same theme I want to bring back is what happens when you run out of options for triple-class plus refractory myeloma. We need more options. We keep needing more options. And so one thing that we are very excited to open as a new option for our patients is a clinical study evaluating a CAR T as a phase 2 looking at GPRC5D as a target. And we will hopefully open this in maybe second quarter of next year. And what I'm showing here is phase 1 data from the same product that was presented this past summer, showing that they were doing a dose finding, and this is just the schematic because it's nice to look at figures. And this is a standard CAR T where it's autologous. We collect it from the product from the patient. As Doctors Callander and Schmidt alluded to, the product is then, the T cells are reprogrammed to target this GPRC5D, and then the product is shipped back to give to the patient. And what is exciting really quickly is that this, even in a phase 1, showed impressive efficacy data, including in patients who had previously received BCMA-directed therapy. And so the point is, we'll have yet another option to offer patients when the novel option no longer works. And what we need is, and particularly in relapsed myeloma and late relapsed myeloma, is always more options. And so I want to conclude by speaking a little bit about future directions in the treatment of relapsed myeloma, or how I see some of the things going. And so kind of same themes even that Dr. Schmidt talked about and Dr. Callander talked about, always novel combinations. We're getting new agents. We're always looking at combining these new agents the same way that, for example, in the SCID protocol that we are hopefully going to open soon. But when we have more agents, you have more choices. And so one question that the field is looking more and more in is, how do you optimally sequence these agents? And for example, if you receive a CAR T therapy and or a bispecific T cell directed antibody, which do you do first, which do you do second? This is a question that we're actively looking at in the field right now, and we're hoping to get more information. And this is the kind of question that's going to keep coming up. Very importantly, is the patient experience. And I think in many ways, to me, this is the biggest question looking forward. As we get more choices, how do we choose the best choice for our patient? Because now that we're having more and more effective therapies that look very similar in terms of how well they work, how do you choose one for the other? What is the best choice for an individual patient? And particularly, as Dr. Callander showed, our patients are, fortunately, we're all getting older, which means our treatments are working better and working longer. But that means, compared to, say, 10 years ago, we're using therapies in patients older than maybe we would have considered using before. And we're learning that we're able to safely offer these therapies, but we have to be uniquely mindful of some of the risks that may be higher in older patients versus younger patients. And finally, in terms of the urgency of response, as Dr. Callander and Schmidt alluded to with MRD, or measurable and minimal residual disease, having an ability to have a more refined finding of early relapse, and then knowing that you're very high risk, and particularly maybe being able to find this from a blood sample and not a bone marrow, so finding relapse even sooner than you may have had before, may lead us to intervene on relapsed myeloma sooner than we ever did before, specifically for the patients that need early intervention who have multiple high-risk genetic features. And so with that, I'll say thank you. And I think it's Dr. Zhang next.
I should.
What? Can you hear me? Okay. Hi, everyone. I'm Jing Zhang, a professor of oncology at the McCardle Laboratory for Cancer Research. I joined the faculty of UW Medicine in 2007. My lab works on blood cancers driven by a family of cell signaling proteins called RAS. It's really my great pleasure to present at the Trillium Fund donor event. This is my first time to be here. You may wondering why, because the early stage of my career exclusively focused on RAS-driven myeloid leukemia. So how did we start in the myeloma? Oh. Why can't I move my slide? That's interesting. It looks like frozen. Yeah. Oh, here. Okay, thanks. The side button. Okay, let me see. I will figure it out. So about 10 years ago, there were two papers published back to back. Those papers actually reported their large-scale sequencing results from advanced myeloma patients. And what immediately caught my attention is they identified about 50% of newly diagnosed myeloma patients actually carry the mutations in NRAS, KRAS, and their downstream BRAF mutations. And now we know that those mutations are not only predominant in the newly diagnosed myeloma patients, they are particularly enriched in high-risk smoldering myeloma, as well as in the refractory relapsed myeloma. So clearly, the RAS signaling is a major driving force for the myeloma progression, as well as mediating drug resistance. So at that time, Dr. Fotis Simokopoulos, myeloma physician scientist used to work at Madison, came to me and asked me if I would be interested to collaborate with him to develop a RAS-driven genetic mouse model. So the idea is kind of straightforward, that you would be taking prevalent genetic mutations in myeloma, and we use a genetic approach, introduce these mutations in the animals. And then we hope the animals will develop similar disease, and that we can use this animal as a platform or as a model to study the disease mechanisms, identify new therapeutic targets, and evaluate how they effectively can treat myeloma. Being said of that, it turns out that myeloma is one of the hardest blood cancer to model in animals. About 10 years ago, the gold standard animal model in the myeloma field is was actually driven by overexpression of a transcription factor called MYC. However, those animals are largely display very indolent myeloma phenotype, and the investigators who generated these animal models over the years trying to transfer the cells from one animal to the subsequent recipient animal, and then selected for lines that can be passage in animals, and then also started to treat them with bortezomib and select for partially response, which means partial resistant to bortezomib. And these actually getting the transplantable V-carbon-MYC lines. However, those lines, they rare acquire RAS mutations. So the field really needs a RAS-driven myeloma model. So here is Fotis. Now he moved to sunny California at San Diego. So indeed, the myeloma field, including Fotis himself, had been trying very hard to generate the RAS-driven myeloma model. Fotis devoted his entire postdoc career of 6-7 years, but failed to do so. So I was very hesitating to take on this project to begin with. He then introduced me to this wonderful lady, Natalie here. I first heard about Natalie, actually it's not at UW, it was actually at an ASH meeting when people learned that I was from Wisconsin-Madison, and they asked me, do you know Natalie? So, you know, at that time, I only know her by name. So Natalie enthusiastically invited me to, you know, seminars, and she's one of the very rare physicians here at Madison that really reply to my emails during weekends. You know, so that's actually a very good sign to me. You know, I finally found a clinical collaborator who was sincerely interested in basic research, not only clinical duties. So I finally decided to jump on board to use my expertise in RAS biology, but I know nothing about B cells, I know nothing about myeloma. So Fotis and Natalie together with, and we also invited Dr. Deming Wang, who's a B cell biologist at a blood center of Wisconsin at Milwaukee. And also jumping on board is Dr. Eric Rainham, who's a pathologist providing us the diagnosis of the animals. So my former postdoc fellow, Zhi, and my former graduate student, Aditi, took on this very difficult and challenging project. So it took them 5 years to combine the upregulation of MYC and a very prevalent NRAS mutation. Together, they created a more malignant myeloma model, which we call VQ. But soon we realized the VQ mice actually displayed very heterogeneous phenotypes. Although they all started with 2 common genetic events, but on top of that, they acquired different somatic mutations. So that's kind of make us wondering, you know, which particular VQ mouse is really represent which group of myeloma patients. So we definitely need a more detailed stratification at the molecular level to look at these animals. So my former graduate student, Evan, took on this task. So he worked with May and Anthony from Milwaukee and did molecular characterization of those VQ mice. So as you probably can appreciate, based on the gene expression profile, you can clearly see that the wild-type plasma B cells group together. And then the VQ actually are divided into 2 groups. The group A VQ actually at a molecular level is very similar as the transplantable V kappa MYC line, whereas we have another distinct, we call group B VQ. It's interestingly that the group A VQ, just like the transplantable V kappa MYC VQ, so they display some of the high-risk myeloma features. For example, they are very proliferative. They are partially responding to bortezomib, and they also spread the tumors into extramedullary sites, lymph node, liver, or muscle. In contrast to those myeloma mice, the group B VQ mice display additional very ultra high risk myeloma features, including that they are expressing the gene signatures that Zhubin just mentioned, SKY92, and the other high risk myeloma features. So this is the current sort of working hypothesis model that we discussed. We think, the group A VQ together with the transplantable V kappa MYC line really representing the RISS stage 3 myeloma, but without ultra high risk myeloma features, whereas the group B VQ mice actually representing the ultra high risk myeloma. And very enthusiastically, Evan in my lab started to delve into, how can we treat those high risk, ultra high risk myeloma mice. So we started in vitro drug screening. We started in vivo validation. In fact, among over a dozen of single agents, we try to treat these VQ myeloma mice. Very disappointingly, only one of them worked. This is a drug called trametinib. It's a FDA-approved MEK inhibitor used to treat the second-line treatment for the skin melanoma. So when we first started to use this drug to treat both group A and the group B animals, so here we're comparing placebo treatment and drug treatment. So you can see the drug actually can significantly prolong the survival of the animals in both groups of animals. But this only happened, we found out later on, this only happened when the recipient animals actually have an intact immune system. They have functional T cell, B cell, NK cell. We accidentally transplanted those tumor cells into immunodeficient recipients. And you can see the drug lost the survival benefit completely. So this sort of steer our attention into the immune system. So in the myeloma field, so you heard from Tim and Zhubin's work, and you know that most of myeloma people really looking at interaction between the myeloma cell as well as a group of immune cell called CD8 T cells. So through the antigen recognition and also mediated by the immune checkpoint ligands expressed on the myeloma cells and their corresponding receptors expressing on the CD8 T cells. So the CD8 T cells can be activated, and under proliferation, they will secrete cytotoxic cytokines, induce the death of the myeloma cells. So those CD8 T cells are the major player in the immunotherapies. However, the consequence of those interaction of the immune checkpoint pathway will lead to the overactivation of those T cells, and they become functionally exhausted. So they have reduced activity, and then they eventually died out. Okay, so that's why in both myeloma patients as well as in our VQ animals, if we treat the mice with immune checkpoint blockade, we did not see significant benefits. So while we're working on the mechanism, we started to realize or appreciate the importance of another type of T cells called CD4 T cells. So in the CD4 T cells, it's, although we call them CD4 T cells, but they have different populations of the T cells. Some of them are helper cells, can help stimulate the activity of CD8 T cells, whereas there are another group of cells are suppressor cells. They actually suppress the anti-tumor activity of CD8 T cells. And what we found out is in the VQ myeloma mice, the suppressor cells actually are greatly expanded. And the expansion of this cell type in the myeloma patients are also linked to the failure to immunotherapies at the moment. So now we're actually looking at how we can develop trametinib-based combination therapies that we can try to kill the myeloma cells, stimulate the anti-tumor CD8 T cells, and in the meanwhile, we can also suppress the CD4 suppressor cells. So obviously, we're trying to do a multi-targeting. This is, I think, is the approach we're going to look for. And I'm hoping that next year I can update you in this regard. So at last, I really want to thank the myeloma research team we built over the years. As you can see that there are many people are jumping on board and working with us. I only presented some of the work we have done. And at last, really thank to the donors of Trillium Fund. They helped me support my former graduate student, Evan, and also helped me recruit my new postdoc fellow, AJ. He will join my lab in 2 weeks. Thank you very much.
Last but not least, we're going to hear from Dr. Miyamoto, and
Yes.
remember the previous presentations. Take it away. Thank you very much. Okay. One question: are we going to get kicked out of the room?
Possibly.
We can try to do some questions outside if that's the case, but.
Yes. Okay. I hope we don't get kicked out by 11 o'clock. We'll see. Yeah. So I'm Shigeki Miyamoto. I'm professor of oncology, same department as Jing that we just spoke. And this is like, I cannot remember how many times I've presented to the myeloma patient groups, and it's always an honor to talk to you. And it's great to see that the progress in the myeloma field. When I started, it was a long time ago, turns out, one of the early Trillium Fund fundee, I guess. So I benefited from that, and I was not, just like Jin, I was not a myeloma researcher before. And Trillium Fund really brought me into myeloma world. And I've been just fascinated and just studying this disease for many years now. And I'd like to talk about a slightly different topic area. And it relates to not cancer cells, but the normal cells. So it turns out the cancer cells have their own, obviously, problems, but normal cells, they co-op normal cells' activities, and then they make the normal cells help cancer cells to become drug resistant. So I want to talk a little bit about that topic area. Oh, good, thank you. I'm gonna try to be within 15 minutes. Okay, so see. So here are the myeloma cells. This is the bone. And myeloma cells live primarily in the bone marrow, as all of you know. And as been discussed, let's see. So there are, broadly speaking, two types of ways that the cancer cells become resistant. One is the cancer cells have its own mechanisms. So they carry genetic aberrations. And then sometimes, as the therapy goes, these genetic aberrations can accumulate further. So they tend to evolve to become more and more aggressive and less drug sensitive. And they can generate their own cell-supporting factors, such a way that they're much more clever at surviving. Okay. But also we know that there are other factors present in the bone marrow. Those factors are generated from normal cells, actually. And because the normal cells, they're not genetic, they're not mutation-based. These normal cells are co-opted, sort of tricked by cancer cells to generate factors. And those factors then feed into the cancer cells, keep them viable. So they become more resistant. So, so far we've heard a lot about the cancer cell intrinsic mechanisms. So I'd like to tell you one story about extrinsic mechanisms that we've been studying for a number of years. Some of you may remember last time I spoke, I talked about our efforts to develop a sort of in vitro assay system in which we try to take the cells from patients and then take the cancer cells and non-cancer cells. So on the left, what you see is the typical histological section where you see cancer cells pointed by arrows and then other cell types. Okay? Most assays to test the efficacy of drugs using patient cells typically utilize only cancer cells. And what we wanted to do was to incorporate these normal cells that are tricked by cancer cells to see if they have impact. And working with bioengineering team, Trillium Fund, who was my former graduate student, they developed these so-called micro-scale devices, and shown here the kind of relative sizes compared to quarter here. So it's a very small devices. And by placing both cancer cells and normal cells and measuring the effects of the drugs, what she found out was that when you put cancer cells by themselves, she was unable to correlate with the clinical response. So in this case, we're using bortezomib, something that we heard already multiple times. But if you incorporate the normal cells in the assay system, the response of the tumor cells will correlate much better with the clinical outcome. Okay, so we were really excited about this. Trillium spearheaded, she patented this technology. She formed a new company called Lynx Bio. So she's the founder and CEO. Now she has a really talented team of scientists working in this a biotech sort of startup company. Now it's also in San Diego. She got smarter, like many folks mentioned earlier. So she's now working with multiple major pharmaceutical companies and making this device into a clinical product. Okay? So from a small Trillium Fund that we received, here's one company that got developed. In the meantime, our lab, because she's spearheading the actual commercial development of the device, we wanted to understand what are the actual factors that are contributing to changing the behavior, drug responsiveness of these cancer cells. So this question resulted in identification of a new factor, something you probably never heard of. In fact, we were very surprised to find this. It's called protein called HAP-LM1. It's a hyaluronan and proteoglycan linked protein 1. And it's sort of a structural view of that protein shown here, kind of a 3 globular domains. These little blue areas, the sort of part of the molecule that's working. There was no prior role in multiple myeloma world. So there's nothing was known about this protein in the multiple myeloma field. We know that it's not expressed by the cancer cells, but it's expressed by other cells, in particular cell type called bone marrow stromal cells. And these cell types, this particular cell types have been studied in myeloma field for a number of years. And they knew that these cells can provide sort of hiding place for cancer cells. You've heard about the minimal residual disease. So sometimes people think that cancer cells are hiding by interacting with these type of cells from drug effects. And I'm going to show a little bit of data on this one. We published a series of work showing that this factor can cause what we think is multi-drug resistance, the number of currently used clinical drugs. It also causes myeloma cells to adhere to endothelial cells, migrate, and home into bone marrow. And we think this is a very important feature of this particular molecule. So it does two main things. One, cause drug resistance. On the other hand, we think it's making myeloma cells basically metastasize to different bone marrow sites. So here's an example of an experiment where we took a mouse and we grafted human myeloma cells to make myeloma tumors. And when you do not use this R factor, this HAPM1, and if you just use bortezomib twice a week, sort of like clinical treatments, and the tumor disappears over time. And showing here the pictures of what they look like in midpoint, about right here. Tumors are becoming very, very necrotic, this is yellow. And then by the time at 22 days, the tumors are pretty much gone, okay? But if you now add our factor, okay, this is not coming from tumor cells. This is coming from normal cells. All of a sudden, tumors look very happy, well vascularized, and then they grow. They keep growing, okay? So it's causing drug resistance. Not coming from tumor cells, coming from normal cells. And it turns out that this factor can also cause resistance to a number of clinical drugs, such as ixazomib, dexamethasone, lenalidomide, this we already heard, melphalan, doxorubicin, bendamustine, but interestingly, not for carfilzomib and selinexor, okay? And I'm going to come back to this one. This is all done in vitro studies. And as I mentioned a little bit earlier, it also causes adherence to endothelial cells. So how myeloma cells migrate from one bone marrow to another site to give you multiple bone marrow features is by myeloma cells entering into a site crossing through the blood vessel and then into the bone marrow site where these bone marrow stromal cells exist. So they're making a HAPM1 and it's making myeloma cells to adhere. Usually myeloma cells might be floating around in the blood, but then they adhere to the myeloma endothelial cells. It can migrate through a very small pore, so they can squeeze in through a small pore, and that this factor can make them do that, and then home into a new bone marrow site. So we think this has potential implications in multiple myeloma cell spreading to different bone marrow sites. Okay, so based on these, we hypothesize that maybe this HAPM1 is a kind of a new non-cancer cell based therapeutic target and possibly some kind of a diagnostic marker. So we asked, does the level of HAPM1 relate to patient outcome? Okay. So it took us quite a bit to develop an assay to quantify this protein, but we were able to look at, in the context of newly diagnosed myeloma patients, it's shown about 26 patient samples. And what we found was that if you have a higher levels of this with a standard of care, you are getting poorer outcome compared to when you have lower amount, shown blue, okay? So clearly it made some difference, okay? And moreover, even though the follow-up time was for the high group, this group was actually much shorter. The number of therapies those patients had to take was much higher. Okay? So what does that mean? Well, we think what's going on is that this factor is causing tumor cells, again, not coming from tumor cells, coming from normal cells, making tumor cells more aggressive. And you need more therapies to treat them. And outcome of that is that you have a large number of therapies that one has to withstand. Okay, so where are we going with this? We think this is a new therapeutic target. So we are working with certain companies to develop an antibody-based therapy. This is an extracellular factor. So we think we might be able to target this by using antibodies to neutralize the activity. And then the second part is that we are trying to develop a detection assay much more sensitive than what we've been studying, so that the hope is, as was discussed earlier, we like to go to peripheral blood instead of going to bone marrow to detect this. So that's our goal. So we're using this target and developing an assay to detect them much more sensitively. Much more sensitively. And we're trying to see whether a high amount of this could be used to guide the therapy towards carfilzomib or selinexor that Zhubin talked about. Okay. So like everybody else, this has really been supported by Trillium Fund, shown on the bottom. So really, that's where it started. And we are funded by multiple different agencies along the way. And the work that I talked primarily about, the drug resistance work, was done by Miley. And Jerome is the one that actually originally discovered this papilloma protein in myeloma samples. And then Shelley did a lot of the drug effects as well. And then Hayen did the most of the work that relates to the migration and measuring of this papilloma protein. But as you can see, there are a number of other students. In fact, Stephanie currently is she's a faculty member at WashU. She's the one who started this under Trillium Fund many years ago. I think Natalie will remember, perhaps 15 years ago, maybe more, quite some time ago. And then again, as Jing mentioned, Natalie has been just an unbelievable collaborator. Without her, I think we would not be able to do any of this work. There are a number of other team just keeps increasing. But I think I want to really thank all of you because I think two really key components. One is obviously the monetary donation, which has been really important to get these young scientists to get going in this field. So we brought a number of scientists into this myeloma fields, which has been great. And then also donation of your samples. Without your samples, we cannot do research. So it's really, every time we get a sample, we're really appreciative of the fact that you have to go through these process to give us a sample. So really appreciate and thank you for that. With that, I'm done. Yeah.
Okay, well, no, thank you so much for all your attention. Just to kind of wrap things up here, I hope that you feel as inspired as we do every day we come into work, because for several reasons. One is, we have the ability to work with obviously these amazing people to think about what we can do for myeloma in the future. And this is just another picture. There's so much we can investigate at this point, so many different ways to go. And science can be slow. As Dr. Miyamoto said, we started talking, I think, back around 2007, I suspect, when that happened. And some of the work, it only happened because of his doggedness and willingness to follow the threads where they go. This takes years, and it really takes dedication, but it also takes, I would say, with my collaborators here, a certain amount of brilliance on their part to figure out how to do this kind of work. So I do think that we are very proud of what we've been able to do here at UW. Our goals for the future that sort of overlapped is we want to continue to have the best clinical trials here for everybody with myeloma. We want to figure out, why does myeloma do what it does, with what Dr. Zhang and Dr. Miyamoto were talking about. And we also want to, for each and every person, provide the best possible clinical care that we can for their myeloma. So I'm going to stop there, and we can have all of the speakers come up if you've got questions for them. Apparently we have this room till 1 p.m. So if you have 2 hours of questions, I think we can handle all of that. So why don't you all come up here and see if anybody has anything they'd like to ask. Yes. Yeah, so the question was, what about blood tests for myeloma? Who would like to, Dr. Schmidt?
Yes, so it's very much investigational in terms of making this a part of routine clinical practice. And there's a lot of tests and a lot of information that we get from a bone marrow biopsy. Some of that, we need the bone marrow. I don't want to suggest that we'll never need bone marrow biopsies again, but we certainly would like to be able to have tests that don't involve going into the bone marrow to assess for things like low levels of myeloma. And so what I would say is, in terms of what I was alluding to in the clinical trials, these tests exist that we could send to try to detect MRD in the peripheral blood, but we don't yet know whether these are good enough. We don't yet know whether we can make decisions based off of this information. And the only way that we'll really figure that out is by testing it in a trial type of a setting. And so I think we're really years away probably from having this be a routine part of practice. Oh,
But just apropos of this, there's an investigator here at UW, because of all the work we've done, who just approached me a couple weeks ago, who's interested in working on a blood test in multiple myeloma to see whether we can come up. This is a gentleman here who's working on what are called circulating tumor cells and has some techniques in solid tumors and is interested in talking with us about multiple myeloma. So hopefully we can. Right now, as Dr. Schmidt said, at least some of the studies have required lot of blood to get enough cells to look around. So it's not, it's quite the donation. I'll just put it that way right now. So, but we're hopeful that we'll have something perhaps here homegrown in the near future. So. Oh, yeah. Okay. Anybody else have a question? Yes, way in the back.
Thank you for your question. You raise an incredibly important point, and one that we still deal with to this day is missed or late diagnosis of myeloma. And so one of the aspects of that we have been working for, and you may have noticed Dr. Callander's involvement in the smoldering myeloma study, is recognizing that myeloma has an earlier precursor state. And at least half of myeloma patients will know about the precursor state before the diagnosis, MGUS, going to smoldering, going to myeloma. And so one of the efforts that we've been working on here is fostering the relationships with primary care providers, basically making sure that they have an easy way to get in touch with us if they are concerned, and also to help guide that testing and make it not a barrier. Now, on a more international scale, there are studies, including in the country of Iceland, that are sort of trying to determine what is the best way to follow your precursor myeloma stage, and even should you be screening the population at large. And I think, again, it always feels like the answer to this question is always soon. Well, it's never, it's never soon enough. And I recognize the needle always feels like it moves too slowly. But we're hopeful, because right now the challenge is we know that screening necessarily probably the entire population, putting everyone through a bone marrow biopsy doesn't make sense. Putting everyone through a full body scan doesn't make sense. We know sometimes based on certain laboratory values that we can get from blood draws that certain people are at high enough risk for myeloma that we should look sooner rather than later. But we don't yet know, should we do that for everyone? Who else should we be looking for? And also, even amongst that, the challenge still remains that even if you are able to find a higher population, a higher number of people to screen for, you're still going to miss some. And I think that's still the part that we have to work at. And perhaps some of the technologies that we've alluded to, such as circulating tumor cells, or sort of maybe at some point there'll be the ability to, at a more sensitive level, really identify who's at risk, who should we look for. But I think right now, I think the ways forward are, one, continued and sort of more further engagement with primary care providers. And that includes us speaking to them sort of at their educational conferences about, these are signs of myeloma. This is precursor myeloma. These are the people with precursor myeloma that you really should look for. These are the risks of precursor myeloma turning into active myeloma. But also, we're going to need either better data in terms of who's really at higher risk, but frankly, probably some better modality, because otherwise there's still going to be people that the myeloma is diagnosed when they show up with a bad symptom, or that they've had, the story is of course like pain, back pain or something. How long does it take someone to do the scan to find that we have and go ahead. No, I thank you for that comment as well. And I mean, I was a primary care provider at some point. All of us that go through this training, we're primary care providers, and we still like to think we provide some primary care to our patients who have multiple myeloma because we see you so frequently. And that is a challenge. And unfortunately, I think the best that I would say is some providers listen to their patients more than others. And, our health system is a bigger discussion than here in terms of barriers and restrictions and sort of this ongoing impetus to see more patients and sort of not just sit and listen. And that's the fundamental thing is Osler said, is that your patient is telling you what they have. You just have to sit and listen. And I mean, I think from my perspective, at least, just trying to remember that each time I walk into the room is an important part, but there's not an easy answer, unfortunately. And if it was, then obviously, we wouldn't be having this conversation.
I actually want to make one comment about this, and we don't have our colleague, Dr. Brunner, who's our informatics person, but this is actually one area that AI, artificial intelligence, may actually help us in the future, because we're basically puzzle solvers, all of us, to a certain extent. You're looking for patterns that you can recognize, but we all know that there's lots of things that you can miss just as you're applying to. And even though I'm not wild about Alexa watching like what I'm eating for dinner or what color my underwear is, but I do think that this may be something in the future where, we can have these big data sets or information help us figure out exactly what you're talking about. So I am hopeful that this will be something in the future.
Now, I actually think that that's a great application potentially of AI and one potential, solution moving forward. I guess, from a what are we doing now perspective, to try to address this, I think what Dr. Gahvari said about education is certainly key. But another thing is that we are growing. And, as we continue on our efforts to try to become a center of excellence here for myeloma and develop a team and, all of that, we can educate our Cancer Connect, who gets our referrals in and get patients in as soon as a potential problem is identified right away. And as we continue to do this and grow and expand, we can make these processes easier for everybody. So I think we're really excited about that growth that we have, and potentially that'll help to alleviate this issue as well.
Any other questions about?
Doctor, you suggested that 3 drugs out of 2 in your trials, and then maybe we'll be better than 3. But there's another possibility, that is that the 4 would replace one of the 3, or 2 of the 3, or all of the 3. Are these considerations taken into account? Do you have trials like that? There are too many trials to do.
Thank you. And that's an excellent question. And so yeah. So the question, if I may paraphrase and correct me if I'm wrong, is, during my portion of my presentation, I alluded to, well, we have trials showing that 3 drugs may be better than 2, 4 drugs are better than 3. Are there situations where maybe 1 drug should replace the 4th drug, should replace all of them? Or are there situations where maybe you don't need all those drugs? And are we doing trials to look at that issue? And I think that's a fantastic question, because now that this is a question session and I have till 1 o'clock to answer the question, I can give a little bit more background. And so one of the downsides to number of the trials that I showed there is they were all kind of unfair comparisons, in the sense that when you have 1 trial that shows AB and another trial that shows AB plus C, generally the AB plus C is going to work as well as the AB and probably better. And if you're a pharmaceutical company, obviously your incentive, and I don't want to, they're incredibly important partners, but an aspect of this is their trials are always comparing a new medicine versus a standard of care, not comparing like it's like AB plus X versus AB, not AB plus X versus AB plus Y. We have many new therapies that we're not getting cross-compared. And this has been a growing recognition that you are not answering these relevant questions. And so how do you sometimes answer these relevant questions? So one aspect is the study that you saw Dr. Callander and Schmidt showed, which was this ECOG study, E181, which is looking at, okay, we know that we have multiple active agents, daratumumab, lenalidomide, bortezomib, and dexamethasone. When do you need all 4? In some situations, is that 4th drug really necessary? And so through a cooperative group, which is, more academic institutions sort of trying to answer this question itself, because no one is, we're trying to answer those questions about when do you really need the 4th drug? When does it add something? Because, I come from the newer era of myeloma where we have all these medicines, but if you go back in time, I mean, from when maybe Dr. Callander was treating patients, they're patients that, that sounded great. I'm gonna hear that. But there are people that clearly sometimes 1 or 2 drugs is incredibly active. There's people that have gotten an autologous stem cell transplant, no maintenance, and are still, have very long-term remissions. There's not everyone that needs more therapy. Some people do just as well as less. And I can tell you at some of the large international conferences, at least other leaders are sort of saying the same question. We need to design trials that also look at de-escalation. With better sort of risk stratification should come sure you should, and you saw in some of these sort of MRD sure, if you have minimal residual disease, as Dr. Schmidt alluded to in, Master 1, Master 2, who gets escalated, but who also gets de-escalated. And I think, to answer your question, yes, we need to ask and try to answer those questions as well as we can. And go ahead. And so to repeat the question out loud, some would suggest that if you have only one drug versus 2 or 3, you may increase the chance that the cancer cell will develop mutations to outsmart the therapy. Were you?
No.
Okay. And I mean, that rationale, for example, is provided by those that would say, let's do a multi-drug combination versus a single drug combination. That hypothesis is why you do sometimes multi-drug versus single drug combinations. If you look back in oncology, sort of agnostic from the cancer type, there are studies that are saying, in some cancers, synergism doesn't necessarily exist. If you have drug A and drug B, the reason that the therapy is more effective is that some proportion of your cancer cells will be responsive to drug A, some will be responsive to drug B, and it's just a probabilistic matter. But we do sometimes, for example, see in myeloma that adding an agent to another may sort of reduce a therapeutic threshold, and there can be a synergistic effect. And I think the answer to your question, though, is there's very much, it depends. Obviously, you saw Dr. Miyamoto and Dr. Zhang. We're some people are looking at myeloma from a very cell-specific fashion versus those of us that are more clinical are looking at the whole sort of everything at once. And somehow you have to meet in the middle and find a way to sort of assess what's happening in the individual myeloma cell or the microenvironment. It's not always easy. And to really answer your question, that's why we do the trials. In some situations, that philosophy holds. I think in other situations it doesn't.
But I think, this is also underscores what Dr. Zhang is doing and Dr. Miyamoto are doing, is that, it's not just the myeloma cell. There's other people around there. The myeloma cell doesn't just grow up in a vacuum. So we have to worry about what those other cells are contributing, what drugs would they be responsive to. And I think what Dr. Zhang's model shows is that we may be even wanting to think about repurposing drugs that we might have otherwise thought for other types of cancer, but we have to think about applying them in different ways. But it's complicated. I think that's what we would all say. I don't know if you all want to add it.
That's good.
Yeah.
So I'll just repeat the question for those listening online is, what is the future of targeted therapy and where we go from here? And just to kind of clarify, we're talking about targeting specific mutations. Is that? So the example that has been brought up and has been the most successful is a subset of multiple myeloma with something called translocation 1114, which is about 15 to 20% or so of patients with multiple myeloma. And this particular mutation is very sensitive to a small molecule, a targeted blockade of something called BCL2 with a drug called venetoclax. And we see very, very good responses with this drug, but only in that subset. And targeted therapy is something that exists in oncology in a lot of different areas. Some fields of oncology, it's all about targeted therapy, and myeloma really has not had as many of these advances. So where do we go in the future from this? I think it's hard to say. Most of these efforts have shown a lot of promise when we take a look at things under the microscope in a petri dish or in mice, and it's been hard to translate that into humans. The most effective medications that we've been able to use are those that are targeting the surface markers and the things that are a part of plasma cells that all plasma cells have, whether they're cancerous or non-cancerous. But we are making progress. I actually think one of the potential driver mutations that might have some sort of a promise, although we're still a long way away from it, is exactly what Dr. Zhang is working on, is RAS. And this is an important molecule in terms of myeloma cell biology. It's upregulated in more than half of patients once they get to the relapse stage. And we're starting to see some inhibitors in other malignancies. And so it's a long way away. And I think figuring out what that specific molecule is that is driving the disease has been it sounds easy to come up with that, but it's a lot harder in clinical practice to get it to work. But we are making progress there. And there are actually kind of along those lines, sorry, I'm rambling here a little bit, but venetoclax is, an exciting drug that we have to offer some patients, but we are actually participating in a trial using a new BCL2 inhibitor, one that might be a little bit more specific and potentially could even be a little bit stronger than that. And so this is something that's currently open here for our patients with 11-14 myeloma in the relapse setting. And so, we're really excited about not just knowing what's going on in this space and pioneering other roads, but also how do we move the targeted therapy that we currently have forward and make it better. Yes, absolutely. It is hard when we start getting subsets and, low numbers of patients to figure this out on a big scale.
I just want to add one area where every cancer treatment is having problems is a very common phenomenon that occurs sometimes at the beginning, but over time, is people lose tumor suppressor genes. And while we come up with drugs very easily shouldn't say very easily, but drugs that block something that's too much, if you're trying to find a drug that actually brings up a level of something that should be there, that's actually a lot harder. We have one drug that we alluded to a little bit, selinexor, that is trying to sort of help restore some of this tumor repressor activity, but that's a big area that needs to get better, I think.
Quality of life and side effects. So a lot of you have been talking about the possibility of the stem cell transplant becoming replaced with other therapies. Can you talk a little bit more about that in terms of quality of life and side effects?
Do you want to start or
Start?
Okay, go.
I think that's a great question, and to repeat for the audience. So quality of life and side effects, and thinking about, for example, the question of a stem cell transplant. Does every patient need to go through with this, and how should we weigh that with patients' quality of life? And one of the interesting things is every conference you go to in myeloma, there's going to be a quote unquote debate. Should we still do a transplant or not? And Dr. Callander recently participated in one of these debates just the last month. And I think, there's always this kind of, it's easy to go up on the microphone to say, we want our patients to live as well as they can for as long as they can. I mean, that's certainly what we obviously want, but there are inherited trade-offs that sometimes we have to go through. And so the stem cell transplant, in a sense, crystallizes that the most, because that's the most intense therapy we have, and that kind of requires the most for a fixed duration. But, my answer, which isn't really an answer, but sort of more of it's just how I think of the question, is that's also a fixed duration of a treatment, at which point you recover. And granted, now we added maintenance. And so in a sense, the intensification more quickly draws down your therapy. And one of the exciting things about CAR T, for example, is right now it's designed to not have maintenance therapy after, which is a benefit potentially for quality of life. You know, lenalidomide is not easy for everybody. And one of the things that sometimes I cringe at is, you know, at some point we're going to have studies that say, well, you add maintenance after the CAR T, well, probably it looks better. Well, then how about our patients that previously didn't have to be on anything for a period of time? And I think getting where we really need to look at more is more of these questions of time limited therapy. You know, who can we sort of give treatment breaks to? You know, I think we've moved away potentially a little bit from that we need to move forward to. And we need to start looking at things such as really just more general opportunity costs of our patients. What are they giving up by choosing a therapy versus another, given that we have more and more options? And really even just the time toxicity of what the treatment will entail. But I'm sure you two will have comments as well.
Yeah, I think that's an excellent question, because one thing that we are trying to get input from people such as yourselves in the audience here is, okay, if we give you one of these new strategies and say, all right, we're going to treat you for a year or some months, and then now we're going to stop. Now we know your myeloma is going to come back. How do you feel about that? And I think we could all say, probably from practical experience, there are some people who would say, that's fine. I can deal with that. There are other people who feel unbelievably insecure in that situation and will say to us, I want to be on whatever you can put me on right now that's going to keep my myeloma under wraps as long as possible. And those are real dynamics here, I think. And I do recall for one of our trials where we absolutely stopped treatment, some years later, this person's myeloma became more active, and that individual said to me, oh, I didn't even realize that myeloma could come back, and so I think we may very well be able to try to offer people something like that, but I think it's not going to be necessarily what everybody wants. But quality of life is certainly something that historically wasn't built in all of these very big trials. It wasn't considered at all, whether some things are just really difficult for people. In terms of stem cell transplant, the quality of life data is actually mixed. I mean, there are some studies that have suggested, they all say the same thing. They all say that 3 months after a transplant, definitely quality of life goes down. The most recent study actually shows it probably improves better over time compared to just standard treatment. But I think that that's something that we need more input from everybody here in the audience. Are you willing to say, all right, I want to be off, but I know that at some point things can come back. I can live with that uncertainty. Or does it feel better to say, okay, we're going to keep you on something that we think is, or that we're pretty confident that's going to delay a relapse based on all the data we have?
I think these are all excellent points. I will just kind of add in from another perspective is that this question of do we still need a transplant has been going on for a long time. So, I mean, the benefit of transplant was first shown back in the late 80s, early 90s. And since a lot of these newer developments that, we alluded to in the first talk, a lot, since these have been coming into play, well, every time something gets approved, the question comes up, well, now do we still have to do a transplant? And, this study has been repeated multiple times. And over the course of the years, it has consistently shown that transplant keeps the myeloma under control for a longer period of time. What is new now is that perhaps we have the tools to figure out which patients are going to do really, really well, regardless of what we do. And could we potentially avoid a transplant in those particular patients? I think that's the first question to ask before we start applying this broadly to a lot of people. And so this involves a better idea of what are the risk factors, a better idea of can we detect disease under the surface with MRD, and also having better drugs that might allow more people to get there. And so I think we're close, but it's not necessarily that we're quite there yet, but that's part of the reason we're so excited about these studies that we're going to do.
And one other study that we didn't have time to mention is we're going to be a site working with the European Myeloma Network to actually test upfront for if you've just been diagnosed either an autologous stem cell transplant or a CAR T transplant right off the bat, and just see maybe CAR T is going to be better in this situation. We don't know yet, but that's exactly the kind of thing we need to do to try to figure this out.
If I can have actually an additional comment, because I think this is such an interesting question and important discussion. One of the things that Dr. Callander alluded to is, she pushed to get quality of life data in the dramatic study. And there's starting to be more conversations that people are recognizing that this quality of life data that we're collecting, that you who are participating in trials are so dutifully filling out on these surveys is very important, because these, for example, have shown things that even a 3-drug combination leads to a better quality of life potentially than a 2-drug combination, even if theoretically an additional medicine is adding to the intensity, well, that's treating the myeloma better. One of the big challenges in generating or asking any of these questions about quality of life is you're intrinsically biasing, because a lot of the people that we want that to answer that question about which would you choose or why, we have to ask it in a research environment. And there's a lot of people that aren't enrolling in trials or don't have that availability. And a lot of times the only people that help us answer those questions are the patients that are enrolled in the trials or are at the academic center or close to it. And there's a lot of patients that we are never able to really ask that question or haven't yet found a meaningful way to sort of include their voice in sort of best answering that question. And I don't have an answer to that. I think it's something that's going to require some maybe creative way in how we, ask these questions in the future.
How wide the search of stromal cell was? You focus just on one from bone marrow, or you spread throughout the whole body? There are thousands types of cells. What was your search?
Yes. Yeah, so it's a great question. We really focused only on, yeah. So for our research, there are different types of stromal cells in our body. Pretty much every tissue contains stromal cells. So do we search the stromal cell activities from different sources or only from multiple myeloma? And our research was solely on multiple myeloma. We did some of the other lymphoma models as well. And we also saw similar activities in some of the lymphomas, for example, mantle cell lymphoma. But our work has been primarily focused on stromal cells from multiple myeloma patients from bone marrow. So there's a possibility that the activities that I was talking about could be present in other cancer types. In fact, the certainly gene expression profile of the tissues, tumor tissues, have shown that the high expression of HAPLN1 can have a negative clinical outcome for a variety of other cancer types, breast, I think it was like, for example, like ovarian cancer, renal cancer, and so forth. So it might be applicable beyond just multiple myeloma. Does that answer your question? Yeah. So then the question is, can AI, artificial intelligence, can aid in this? Absolutely. I think that one of the things is that most studies have focused on cancer cells themselves. So the actual data on stromal cells from not just myeloma, but other cancer types are relatively limited, because most people focus on cancer cell themselves. So I think we need to collect those data first in a much more wider way, so that we can then ask hopefully intelligent questions to see how relevant or how specific stromal activities might be.
Okay, any other questions? I know I think we all appreciate you all hanging in here for so long. And again, I want to thank each and every one of you for coming today. We're very excited about the future here. And, as Dr. Miyamoto said, we wouldn't be here without just your support, but also your bone marrow support too, that you've been so gracious to offer to us over the years. So we hope that we will be able to come to you maybe next year and give you some updates on these things. Maybe we don't want to wait 6 or 7 again, but thank you all very, very much for attending. And again, thanks for the support.